Welcome everyone, and thank you for taking time to join us for this exciting webinar on nanoparticle characterization from NanoFCM. I'm Dr. Clayton Deegan, the Sales and Applications Manager for North America, and I'll be your host and presenter today. Along with me is Dr. Ben Peacock, Head of Research for EMEA at NanoFCM, and he'll be available to answer any questions you have at the end of this presentation. So without further ado, let's jump right into this webinar on Assessment of LNP Quality and Encapsulation Efficiency by NanoFlow Cytometry. NanoFCM is a global provider of dedicated nanoflow cytometry platforms. We're specialists in nanoparticle characterization and quantification, and recent rapid uptake of NanoAnalyzers has been for LNP measurements, particularly with respect to mRNA therapeutics. So our company's statement, NanoFCM brings reliable and quantitative measurements to the nanoscale, supporting the emergence of new classes of diagnostics and therapeutics. And in today's example, we're going to focus in on the one application space here of nanomedicine, particularly with LNPs and LNP-like particles, and examination of their size distribution, concentration, and cargo. Let's take a brief tour of the NanoAnalyzer. The samples loaded in a standard tube in small volumes is only about 1 microliters consumed during the measurement. The samples injected into a flow cell through a narrow capillary, and the sheath flows under gravity, and at appropriate dilutions, our particles will pass through the detectors as individuals, and down one optics line, we see the side scattering from the particle, inferring its size, and 2 different fluorescent detectors aligned in time, providing true single particle analysis and phenotyping of these individual nanoparticles. And each of these detectors is actually a single photon counting module, which provide extreme sensitivity in both the scatter and fluorescence, which when combined with the slow sample flow rate in the flow cell after the capillary, which increases the particle dwell time in the laser, these aspects combine to provide the level of sensitivity and resolution required for accurate, reliable nanoflow cytometry measurements of many particle types. Let's take a look at some simple data. We've redrawn the detection configuration, and here we're going to look at just one fluorescent channel combined with the side scatter, and we're going to detect the intrinsic fluorescence of the API inside some liposomes here. So when we look at the middle 2 traces, we see a typical side scatter trace where there are individual bursts as particles pass the side scatter detector, and the height of each of those bursts infers their size. Then in the bottom trace, we see the same time frame, the fluorescence detector, where individual fluorescence signal is triggering in time, shown as those individual bursts. And what the system does is aligns these in time so that it identifies those particles which are containing the fluorescent signal, and then plots them out on the right in the scatter triggering mode, where all of the particles in the top trace are plotted along with their intensity in the channel that's being detected in fluorescence. And here we find that 42.9% of these particles contain the API in a simple analysis that detects them based on their presence as a particle, and then says, is that particle positive for the cargo? This type of single particle analysis provides true multi-parameter LNP characterization, starting at single particle sizing and concentration without labeling. Because we can detect all of the particles without labeling, the opportunities presented for percentage of those LNPs that are loaded, that have a characteristic that identifies them from all of the others in the mixture, be it cargo or a surface characteristic or a targeting moiety. The measurement is sensitive enough to quantify these loaded materials, quantify the surface density of a targeting moiety, quantify the cargo. Simple, intuitive experiments using things like RNases or DNases can identify cargo locations in particles. Is it surface association, or is it protected inside the particle from the activity of some of these enzymes or other proteins? And then because it can generically sense particles or sense fluorescently labeled things that are smaller than the scatter trigger can see, sensitive to contaminant material, things like nucleic acids being outside particles or independent of particles, things like empty particles, or other things that may be present that aren't even related to the formation of the LNPs, but ride along through the formation and purification process. take a more in-depth look at each of these characteristics of multi-parameter LNP characterization. Starting at unlabeled sizing and concentration. When we have a sample, we can simply dilute it into an appropriate buffer, inject it into the machine, and recover the number of particles that are present, and their number-weighted size distribution is shown here in the blue trace. When we look at the sizing in nanometers, it's based on the scattering intensity like we saw in previous slides, and what we use is calibration tools that we provide. We see here our 4-point calibrator, the S16M EXO standard. These are mixed silica beads that are individually sized at 68, 91, 113, and 155 nanometers. And here they're mixed in even number concentration and detected in the same sample to produce the result we see in the lower right. The height intensity that produces each of those sizes and size peaks is then used to create a calibration curve that results in our blue trace for our sample where its median size is detected. Among the multi-parameter LNP characterization things you want, cargo presentation or encapsulation, and of course subpopulation sizing and concentration. So the easy ability to report only about particles that fall in a specific range of sizes or other parameters that make them your particle of interest. So as a simple schematic, we take the particle, perform some type of labeling technique, and then perform the analysis. And as an example here, we'll look at empty particles exposed to the mRNA labeling procedure. So these are LNPs formed without nucleic acids. And we can sense them in the scatter. The dot plot's full of data that says we have particles, but our mRNA labeling on the y-axis doesn't result in any of them appearing to be positive. So these would all be empty. And when we perform the same procedure in the presence of a loaded particle, the mRNA or other target inside the particle is labeled, and we can immediately sense the size distribution, and also that among those particles we're sizing, the vast majority do contain the cargo of interest. We see that here in now two positive formulations. So we're going to compare two LNP formulations that both have nucleic acids when they're formed. And we see that we get two different loading efficiencies through the lens of total particles. So of the particles we can see in LNP1, 97.7 contain the cargo, and in LNP2, only 90.3% contain the cargo. We can see this also as an expansion of the blue population, or P2, being negative. There's nearly the same number of total particles in these, but the distribution of nucleic acids and the total percentage of particles which contain nucleic acids is different and easily identified here with the NanoAnalyzer using a simple cargo presentation encapsulation assay. So overall with the NanoAnalyzer platform, you're getting single particle sizing and concentration, average size, size range, the standard deviation, aggregate identification, and a number weighted basis, and using standards that let you know the performance of the instrument every day before you start collecting data. You're able to easily detect the loading ratio through simple, routine, intuitive experiments that will show you from the number of particles you have, what percentage are loaded with the cargo you want, the concentration and size of that distribution, and the fluorescence abundance of these different cargoes or APIs, and the quantity of loaded material, mRNA copy per LNP, average range relation to size when it is or isn't loaded, if it is related to the size, and the presentation of other surface ligands or targeting moieties can be easily detected alongside the nucleic acid cargoes and the size of the particle. Now that we've covered the basics of the NanoAnalyzer capabilities, let's put them to work in some applications and case studies and dive into some more complex data and examples. Let's get right into the application LNP users are most often interested in, and that's co-detection of non-encapsulated nucleic acid material, and we'll see what we can learn here. So we've got our dot plot right away, and we've got our mRNA labeling signal on the y-axis and our LNP size on the x-axis, and we might notice right away that this plot splits left and right against the size, and that's because here we're using fluorescent triggering. Instead of just counting the particles and then plotting whether they're empty or full, here we're counting the cargo signal and plotting whether it's inside or outside of a particle. And that leads to seeing the free mRNA here, as opposed to the mRNA-loaded particles and a number of events ratio. That doesn't mean that they're the same intensity. It means that the number of them relative to each other is around 3 to 1 here. And when we look at our detectors, what does this data coming through the detectors look like? On the scatter detector, we see the particles. So all of the particles on the right in the mRNA-loaded family fall into this group. They trigger the side scatter detector, and they also have a coinciding fluorescent signal. Both detectors are well resolved here. We can see each of the events as an individual in time, and we find that there are signals in the fluorescent detector when we're in fluorescent triggering that don't coincide with a particle, as we've indicated with these sets of red arrows. Those events show up in the blue trace as free mRNA or nucleic acid cargo that's outside of a particle. We can zoom in further on this by running non-encapsulated mRNA labeled with the same dye, and we see these peaks right around the same height. The scale zoomed in. Our particles were at 600 to 800 in height, and our small little peaks were down around 100, and our non-encapsulated mRNA in solution right here between 100 and 150 in intensity heights. Those are our pieces of mRNA, and we can use this information further for quantifying the LNP mRNA loading. So now we're going to plot in our first dot plot here both the free RNA, what we're called the vaccine, the loaded particles, and our non-loaded vaccine, the small blue family at the bottom. And we can overlay those for their size distribution. The blue population is less numerous and smaller, so it shows up as a little peak, is 60 to 80. And then our loaded vaccine is in the red distribution. And here we've done the same thing, labeling mRNA with membrane permeable nucleic acid dyes, and we're utilizing both triggers. And then we're taking that fluorescence intensity of the single mRNAs like we saw in the trace we just showed, and we see those peak burst traces right around 100 height and intensity. And we use that value to determine the intensity of a single RNA cargo. And then we can take the intensities of the individual particles, which are part of that red distribution or vaccine trace, and we can look at how many copies or how many fold higher is the signal in the particles than our free RNA. And we come up with this frequently 10 is the most frequent copy number, 10 copies per particle, and they extend all the way out into the 30, 40, 50 copies in single particles. And so in this way, we use the fluorescence and scattering trigger modes both, along with free RNAs, to determine how loaded our loaded particles are. Okay, so we've taken a look at how loaded particles are. Can we determine where they're located or how their loading has gone on and their cargo location on these LNPs? We'll have an example here. So we're again going to exploit this membrane permeable nucleic acid label, and we show our dual triggering here. We've got our free RNA, our loaded particles, and our empty particles in the yellow, red, and blue respectively. When we use a membrane impermeable nucleic acid dye, we greatly increase the blue population, or those that are apparently unloaded. We notice, though, that we haven't eliminated the apparent red population or loaded particles, even when we're using an impermeable dye. And we certainly still see the free RNA in the gold population because it's outside of a particle. When we add the RNAs to this solution and we digest those RNAs which are outside of a particle, not protected from the RNA's activity, and then use a membrane impermeable nucleic acid dye, we find out that we eliminate that signal that was coming from the impermeable dye in the formed particles. The free RNA is removed, and a lot of the RNA that was associated with the particle signals in the presence of RNAs and an impermeable dye is digested by the RNAs. So you can utilize these types of experiments with processing the cargo to determine whether it's associated with the surface or whether it's actually protected inside the particle, and to determine whether what you see to be free cargoes are actually free or also protected. or my favorite examples, a dual fluorescent reporter assay along with sizing, and we're going to see some really interesting features of the particles under exposure to different chemistries. So this is going to be a fluorescent analysis of C11 Bo-Dippy modified liposome. So the C11 Bo-Dippy molecule will intercalate the liposome lipids, and under exposure to reactive oxygen species, or here peroxide, it's going to shift its fluorescence from orange to green, and we're going to see less intense orange and more intense green under exposure to reactive oxygen species. At the same time as that goes on, the reactive oxygen species should also cause the efflux of the intrinsically red fluorescent cargo molecule MXT to leave these single particles. So under exposure to ours, we're looking for their redness to become less. They'll become less intense and red as the MXT drug leaves them. And what does this data end up looking like under exposure to the nominal buffer and exposure to the reactive oxygen species? Exactly what we just discussed in the side scatter burst trace. We see the size and RI decrease. In the green fluorescence burst trace, we see a big shift to green intense as the C11 Bo-Dippy reports its green shift in reactive oxygen species, and we see the red intensity shift down as the drug leaves the individual particles. So this is again through the scatter triggering. We detect all the particles. We secondarily detect that they are green and or red, and how green or red they are is used in between the two conditions to infer the change in their size is related to the change in their membrane and is related to the change of the drug leaving the particle. So this is a perfect example of a multiple fluorescence and size exploitation to do a simple experiment to learn a lot about the behavior of these particles under exposure to reactive oxygen species. So we've looked at a couple of examples already that deal with LNP cargoes and their detection. Let's here look at surface modification of particles and detection of ligands that are supposed to be displayed on their surface. And we can think of this just like we might think of a cell. In the particle, it has a certain number of targets available. Different formations may lead to different display of them on the surface and their evenness. It may lead to different numbers. And what we want to inspect here is through using a interactor with that desired molecule, can we detect its presence on the particles just like we're doing with the cargo? What percentage of the particles have it? How bright are they relative to each other? And then in turn, can we determine how many ligands that is? So this is a surface folate bound by a fluorescence-linked folate receptor. So the particles are formed with the folate on their surface. And then we can right away tell that the folate's configured in a way it will react with receptors because we have our fluorescent-linked receptors reporting back that they're labeling the folate conjugated liposomes. The fluorescence intensity is relative to the number of folates per liposome, and we see a nice little curve here against the distribution of the particle size and the distribution, therefore, of the fluorescent events. So it seems related that the larger surface area particles have more folate molecules entrained in them. How many is that? Using the size and count data for the number of particles, along with the intensity of the folates, we arrive that there are 0.8 folate ligands per 100 nanometers squared of liposome area. And that type of analysis can be achieved for any type of molecule you ask to be displayed on the surface of the particle that you have an interacting molecule, even like an antibody that's labeled for. You can arrive at detecting different amounts of that on different particle preparations, how many particles in your preparation display the desired target, and how many targets are there per particle. And we've seen a number of great examples now of how the NanoAnalyzer is being used by nanomedicine makers to determine their concentration of total particles and of subpopulations of loaded or unloaded particles, the size distribution measured on a particle by particle basis down to 40 nanometers, and really here, the deeper characterization of those single nanoparticles, the ability to determine protein, lipid, nucleic acid content using intuitive fluorescence detection experiments is the biggest draw of the NanoAnalyzer platform. And then speed, we're getting to this data in a matter of minutes. If we prepare an LNP sample and we label it in 2 to 3 minutes, we have the results for the size distribution, the concentration, and the loading efficiency. NanoFlow cytometry is a powerful and flexible platform for comprehensive nanoparticle analysis, and we would love to hear from you. And in fact, we'd love to hear from you right now, and we're going to have a Q&A here with myself, and as I mentioned, our head of research, Ben Peacock. And so if you've got questions out there, we'll get started here. And I'm going to put our contact information up during this, but feel free to reach out if you'd like to learn more, have a demonstration in your lab, or see what the NanoAnalyzer can do for you. Thank you for your time and attention, and we'll get started with the Q&A.